Hello everyone, and welcome to our webinar on Discovering Nature's Protection with Organic Antimicrobials, organized by IPI. My name is Yaling, Senior Innovation Manager from the Food, Healthcare and Sustainability team at IPI, and it is my pleasure to be your MC for today. Antimicrobial protection has been a key subject in many industries and applications, but the pandemic has definitely increased everyone's awareness on the need for antimicrobial solutions. Together with the drive for sustainability and eco-friendliness, we are also seeing an increased interest in natural organic antimicrobials. With that in mind, we have brought together technology owners and experts from Singapore and UK as our guest speakers for this webinar. We hope that you will be able to gain some insights from them on plant-based antimicrobial agents, antimicrobial peptides, as well as intricacies in antimicrobial efficacy testing. For those who may be unfamiliar with IPI, there will also be a short introduction after this. Before we get started, here are a few house rules. All attendees will be muted during the webinar. Should you have any questions for our speakers, please type them into the Q&A panel. We'll bring them up during the panel discussion later. The presentation slides will also be available for download at the end of today upon the completion of a post-webinar survey. Without further ado, let me invite Alvin Howe, Head of Food, Healthcare and Sustainability at IPI, to kickstart today's presentations and share with you more about leveraging open innovation platforms. Alvin, over to you, please.
Thanks, Yaling. Let me share my screen. Okay, hi, good afternoon, everyone. Hope everyone can hear me loud and clear and see the screen as well. So thank you very much for coming over today and going to listen to this short presentation on IPI. Okay, so just jumping into everything, IPI is basically a non-profit organization, and we offer companies the opportunity to grow beyond their boundaries. So we are also funded by MTI, and have been around since 2011 April. So this year we turned 10. And how do we actually go about helping companies grow their businesses, right? We have an extensive suite of services that enables companies through their open innovation journey. I'll go into details on these particular different services in the next slides. Okay, over here we can see our open innovation services, and these services basically on technology scouting, technology landscape studies. They are very bespoke, tailor-made services that is, tailor-made according to your needs. And of course, if we talk about this engagements with IPI, we are very focused in terms of bringing you the right technologies in order to enable your business. On the Innovation Advisors Program, we actually have a pool of bilinguals, and these bilinguals are actually very senior and industry veterans, right? And they are good at both technology as well as innovation, as well as business aspects of things. And they are able to help accelerate the go-to-market with companies using their knowledge, experience, as well as through their network. On the innovation marketplace, is actually a virtual marketplace for seekers and providers to list their technology needs and offers respectively. Of course, if we talk about reaching out to our global audience of about 25,000 subscribers, we have our weekly tech alerts, which showcases the latest technology needs and technology offers on our platform. On the Open Innovation Challenges, we actually work with various types of corporates as well as government agencies on their innovation challenges. So we have a list of various innovation problem statements, and we'll invite everyone to check in once in a while to look in for opportunities to provide your solutions to these problem statements. For the network access portion, if we talk about how IPI actually reaches out to different areas or different industries or different companies, right? Doing this kind of webinars similar to these sessions, small scale events for technology showcase or networking sessions. These are one of the things that we do. We also have larger scale events in terms of incorporating both technology showcasing as well as networking, such as IPI's flagship technology innovation event, which is called Tech Innovation. Our staff actually works with on various portfolios, and some of them could actually go out in terms of white papers, right? And in terms of workshops, these are more educational in order to provide everyone with sharing our experience in terms of commercializing technologies, as well as challenges in open innovation. And currently, actually, we have one technology workshop currently ongoing in parallel. And also another one that's upcoming will be on the open innovation workshop in January next year. So this is also a snapshot of our global network of partners. We work with various types of partners from academia to companies as well, local as well as overseas, trade associations, government agencies, as well as commercial IP intermediaries. We work with them in order to engage them as well as reach into their network of solutions, whether be it technologies or even other companies, and kind of bring them over to engage with our local enterprises. And nothing beats hearing from companies that have benefited from IPI services, of course. So we have more success stories that you can view on our website at the link below. So why do you want to work with IPI? Of course, IPI, as a company, we offer you neutral perspectives to things relating to technology, as well as offering you other areas of advice from a neutral perspective, right? We also cover a broad spectrum of industries. Our staff used to be from the industry as well, so they can definitely understand your industry needs better. And as you have seen earlier, our network is also global, right? And we frequently tap into them in order to enable technologies and solutions for our local enterprises. So of course, I would like to end off with these last two slides of my presentation by sharing some insights that we have gathered on this particular topic of antimicrobials. So I hope through this session, everyone can actually discover more about antimicrobials, understand it further, and definitely to apply them. So over the last 2 years, we have gathered various areas of interest from companies, right? As you see on the screen right now, we have various interests from different companies in space of natural plant-based antimicrobials. Of course, people also look into surface treatments of various materials and things like that. Also in terms of coatings, to apply the antimicrobial as a coating. So that's pretty common as well. The antimicrobial technology itself can also come as an additive, right? So if you put it into things like plastics, packaging, and you can try to enable that into a kind of functional antimicrobial packaging, right? There's another area that is also very commonly asked about, which is the biocompatibility of the technology. And if you talk about biocompatibility, is becoming more and more of interest these days. And a lot of people, they look into the how it actually reacts with the body, whether there's any adverse reactions and things like that. So this is quite common, especially in the space of personal care, right? And if you talk about the other areas, I will share more with you on where they are actually applied in. So the industries in which antimicrobial technologies are actually applied in covers a wide area. Sanitation, that's pretty obvious. That actually covers about 25% of the inquiries that we actually have. In terms of chemicals, these are companies who look into manufacturing the technology itself or producing these antimicrobial technologies. You hear from some of them later. And of course, we talk about applying to building and construction. These days, this is also pretty common. And subsequently, we move into areas like personal care as well as food, which is quite similar in terms of percentage. These are one of the few common areas that people are looking to apply antimicrobial technologies to. So feel free to reach out to us if you are interested in these areas or even other areas of technology. So we'll be more than happy to reach out to you and also work together with you to see where we can help you. So with this, I end my presentation. So thank you very much. And I hand over the time back to Yaling.
Thank you, Arvin. So next up, for our first guest speaker, we have Dr. Kai-Yang Lin, Chief Scientific Officer of Estar Technology, a Singapore startup, to share with us about his work on next-generation antimicrobial agent derived from nature for broad-spectrum applications. Kai-Yang, over to you, please.
Hi, thank you, Yaling. Good afternoon, everyone. So let me share my screen and we can get started. So I'm Kai-Yang, the Chief Scientific Officer of Estar Technology. First of all, I would like to extend my thanks to IPI for giving me this opportunity to speak at this webinar on some of our development and work at Estar Technology. So since I'm the first of the panelists to kickstart, maybe let me give a bit of introduction, right? With the explosion of the COVID and the issues of superbug coming on, right? There is an increase in demand for antimicrobial compound, right? And the market is really quite big for antimicrobial compound, going up to about 4 billion by the year 2030. Right. So what are some of the antimicrobial compounds that we see in the market? Most of them are chemically synthesized. You have antibiotics that your doctor prescribed, chlorhexidine. Some of the newer technology includes quaternary ammonia as well as silver-based complex. There's no doubt that this kind of chemical compounds are effective, right? But they are also associated with some drawbacks, which includes potential cytotoxicity, chance for microbial resistance. They are mostly unsuitable for kids who have a long life ahead of them. And with the public being more educated and informed, there is a greater resistance from the public towards such chemical compounds. And as such, there is a larger and larger demand for natural antimicrobial compounds. So what are natural antimicrobials in short? So they are biomolecules that we derive directly from natural products such as botanicals, food waste, and even metabolites of bacteria. The market for natural antimicrobials is quite big at about 1 billion, right? So it is about 1/3 of the total market for antimicrobial compounds. So some of the prerequisites that we desire from these natural antimicrobial compounds include them being able to kill bacteria. That's a given, right? They should be highly compatible with human and animal health. And last but not least, their production and manufacturing should be environmentally sustainable. So if you ask me, are there any natural antimicrobials out there in the market? I would say yes, right? We have bee products such as honey and propolis, which are well known. We even have antimicrobial peptides like lysine, which is used as a preservative in food application. But these natural antimicrobials are always associated with certain drawbacks, which include a weaker antimicrobial functionality, meaning you need to have a higher concentration of them to elicit a certain effect. Secondly, their extraction efficacy and yield is quite low, making it very expensive to produce them. And last but not least, because they are natural compounds, they are a lot more vulnerable towards processing conditions such as heat treatment, pressurized treatment, making them unable to be used for downstream applications. So this is where Estar comes in. Estar Technology wants to bridge this gap. So basically, Estar Technology is a biotech company that looks into the research and development of novel, natural antimicrobial agents. The basis or the foundation of the company really looks to explore and screen natural products for potent antimicrobial agents, as well as engineer and optimize extraction strategy to efficaciously extract these bioactive molecules. With the technology being developed, we then move one step further to apply them into different applications as well as products for different sectors, which includes FMCG products, biomedical sectors, or even the food industry. So we are very excited and happy to officially commercialize our first natural antimicrobial technology, the Ultra BacTech, in May this year. Some features of our Ultra BacTech include it being derived directly and purely from plant sources, Secondly, its extraction is 100% clean label, where we do not make use of any chemical solvents or additives. It has a good potent antimicrobial functionality, being effective against both bacteria and viruses. Of course, being natural, it has a good biocompatibility with human cells. Also, because of its origin, the production and manufacturing of this ultrabacter is highly eco-friendly. One key point that I would like to highlight is that the ultrabacter has good processability, meaning they are very resistant towards downstream processing, such as high temperature as well as pressurized treatment. And as such, it can be used for a broad spectrum of downstream application. For example, the ultrabacter can withstand the rigors of a whole autoclaving cycle without compromising its functionality. So here are some testing reports that we have done. Basically, this is a chemical analysis to show that the ultrabacter is 100% natural. It does not contain any chemical solvents or additives, nor does it contain any heavy toxic heavy metals in their compound. Of course, in terms of functionality, the ultrabacter is highly effective against a broad spectrum of different bacteria, which includes biomedically relevant strains such as E. coli, Staphylococcus. We are also effective against spores-forming bacteria such as Bacillus cereus, as well as food-related pathogens such as Listeria monocytogenes. Of course, these days, no antimicrobial compound can be claimed as antimicrobial if it is not effective against viruses. As such, we tested the efficacy against viruses using the EN 14476 test standards. And we passed with flying color, proving that ultrabacter is highly effective against both enveloped and non-enveloped virus. We would also like to take this platform to announce our latest results with the SARS-CoV virus, where the ultrabacter is able to completely inactivate all of the virus loading within a matter of minutes. This translates to a 99.999% killing of SARS-CoV virus by the ultrabacter. So, in general, the ultrabacter kind of ticks all the important prerequisites of a good antimicrobial compound. It is effective against bacteria and viruses. It has good biocompatibility. It can withstand the rigors of processing conditions such as heat treatment and pressure treatment. And last but not least, its manufacturing is highly sustainable. So we have a good technology. We have developed it. What's next? We take it a step further by putting it into different applications and products. As a proof of concept, what we did is we incorporate the ultrabacter and push out our very own line of personal care products, which includes our Estar hand sanitizer as well as the air and surface disinfectant. This product was launched in July this year and is selling very well through both online and offline platforms. Of course, we not only push out our own products, we work closely with business partners who are interested in our products. Gimli Global is one example of business partners where we work with collaboratively. So what we did, we tried to incorporate the ultrabacter into fabrics, imparting a certain type of antimicrobial functionality to the fabric. From there, we make it into different products, including the popular Ultra Mask, which was distributed by the government to the Singapore population during the recent distribution exercise. We also make it go one step further to make it into PPE such as medical gowns, medical scrubs, and even lab coats. So going forward, Estar Technology is really keen to work with more interested business partners to expand the use of the ultrabacter. So please feel free to contact us and come to us if you have any interesting ideas that you would like to incorporate the ultrabacter into. So I would like to finish off my presentation with this slide. Basically, it's a question that I'm always being asked. How does natural antimicrobial compound really kill microbes like bacteria? So there are a lot of hypotheses on how this natural compound kills the bacteria, but one of the popular mechanisms would be the carpet model. So what is the carpet model? Basically, you have a bacterial membrane that is made up of a phospholipid bilayer. When you add natural antimicrobial compounds to it, it gets inserted into the membrane and stretch it, right? As you add more, this natural antimicrobial compounds goes into the membrane and stretch it further. Eventually, the membrane is stretched to a point where it completely disrupts, in the process, killing the bacteria. And this is the carpet model. And this is most likely one of the main mechanisms that this natural antimicrobial biomolecules use to kill microbes. And with this, I come to the end of my presentation, and this is my contact. Please feel free to contact me if you have any question on our products and project. Thank you.
Thank you, Kaya. Our next presentation is from Dr. Don Wellings, founder and managing director of Veritech based in the UK. Due to unforeseen circumstances, he is unfortunately unable to join us live today. Sorry about that, but not to worry. We have actually prepared a recording of his presentation. So if you have any questions regarding his technology, please continue to post them in the Q&A panel and we can address them after the webinar. So now let's hear from Don about the journey from a natural food preservative to an antimicrobial microparticle.
Hello, my name's Don Wellings. I'm the founder and managing director of Veritech in the UK. Back in 2012, I think it was, we first started to look at using an antimicrobial peptide called polyethylene lysine as a solid support for solid phase peptide synthesis. Of course, we quickly realized that polyethylene lysine as an antimicrobial peptide could impart in antimicrobial properties on the microparticles we were developing. So we inadvertently discovered something else. Now, polyethylene lysine, the structure of which is at the top of this slide, is a cationic peptide. This material, of course, as a food preservative is ideal because it's soluble, it goes into foods, it can be mixed with many foods and food preservatives. But if you want to use it in a topical fashion or intravenously or even orally, then it will degrade and it will dissolve very quickly and very rapidly. So to slow down that degradation, it would be a good idea to convert it to a microparticle or to some other structure. The material itself was discovered by a Japanese group back in the 1980s. And very fortuitously, they realized what they had discovered, which was a new food preservative, a natural food preservative. By 1987, this was made on a multi-ton scale as a food preservative in Japan and Southeast Asian countries. So it was approved in Japan. It was actually approved by the US in 2003. So it's now generally regarded as safe. It's used in China and, like I say, many other Southeast Asian countries. Now, the advantage of converting this back into a microparticle is to actually increase its stability either on the skin, intravenously, or orally. So what are spherosomes? Now, spherosomes is a different technology. Again, back about, I think this was maybe about 2010, we discovered this by accident, that you can self-assemble fatty acid microparticles in water. A very simple approach. Now, the technology was already patented and protected. But everything happens in water. The raw materials that we prefer to use are bis-carboxy fatty acids, such as brassilic acid, azelaic acid, sebacic acid. Brassilic acid, as you can imagine, comes from the brassicas in our diet. So these are all natural fatty acids. As I mentioned, made on a very large scale because they're used in the textile industry for making nylons and various other plastics. Now, if you take polyethylene lysine and absorb them into spherosomes and then cross-link them, you end up with antimicrobial, stable, biodegradable particles. These have been tested in many ways in our company and with our collaborators. So we now have a spherosome with this positively charged polycationic antimicrobial peptide on the surface. Interesting thing about these things is most of the surfaces we're interested in are negatively charged. So skin is negatively charged, plant surfaces are negatively charged, work surfaces, cellulose work surfaces, wooden work surfaces are also negatively charged. So this polycationic positively charged peptide is readily absorbed and trapped on the surface of the areas that we're interested in, whether it's skin, plants, or wood or work surfaces. They stay there for long periods, again, because of their size. They get trapped in the pores on your skin, and they stay there, let's say, for long periods, even after washing for long times. These carefully spaced charges, developed by nature really, are very clever in that they're spaced to coincide with the phospholipid surfaces on bacterial fungal cell walls. So they punch holes in the cell walls of these horrible microbes, leaving our cells untouched. There is a good reason for that, but it's too difficult to go into at this stage. As I've said, the technology for spherosomes, as with the technology for cross-linking polyepsilon lysine with fatty acids, is already now protected by many worldwide patents. This is just one example, and this is actually the example on cosmetics. So what do they look like? They look like little balls with little knobbly surfaces, particularly the ones with polyepsilon lysine attached to the surface. So you can see these are all quite uniquely, almost polydispersed, around about 2 to 3 micron in size. So just the right size to get stuck in the crevices on your skin and on wooden surfaces and on plant surfaces. So a bit like sticking Velcro, you can imagine these little spiky balls sticking to the surface of a cell. The actual activity is to rip out a part of that cell wall and break up these microbes into small pieces. So we have a scanning electron microscope picture of E. coli and a spherosome, quite happily separately. Mix the two together, and what happens is you break the E. coli down by penetrating cell wall, spilling out the contents and breaking the E. coli down into small fragments. And this is typically what you see with fungi and with bacteria. A little bit closer up, you can see really clearly on one of those E. coli is the hole in the wall that has been caused by the positively charged surface of the spherosome. So what have we looked at? Many, many bacteria. This is just an example. We specifically have been interested in looking at mixed species biofilms. Bacteria don't live by themselves, as do fungi. They don't swim around, they live symbiotically inside a biofilm. Of course, wouldn't go without checking out to see what they do to viruses. And yes, they're effective against coronavirus 229E, which is actually the common cold, but it's the standard test for coronavirus activity. All of this work that I'm publishing here, that I'm mentioning, was done independently by an accredited company working to ISO 9001. So this isn't work done in-house, this has already been assessed independently by others and also by many universities that we work closely with. So getting the 7-log reduction, 99.99999% reduction of some of these organisms in bacterial biofilms. That's quite an achievement. So we can do lots of things. We can absorb drugs, we can absorb proteins, we can absorb lipophilic materials like oils, aromatic oils, edible oils. We have developed an alcohol-free hand sanitizer called Spherolyze, where the active ingredient are spherosomes with polyepsilon lysine. We also have a wound dressing based on the same technology using those spherosomes called Proliferex. And this is in a clinical study at Queen Elizabeth Hospital in Birmingham, with a hope that next year we will be CE marking that. We've also developed a clear hydrogel and cross-linked polyepsilon lysine, and we have a corneal bandage in a clinical trial that should go into human volunteers early next year, again with a view to CE marking. So as I mentioned, we can immobilize many things, and this is maybe a slight distraction, but we can immobilize hemoglobin. So hemoglobin immobilized in spherosomes gives you an artificial red blood cell, and these have been tested and proven to maintain porcine kidneys outside the body ex vivo. It's important to say that hemoglobin is also highly antimicrobial. So these artificial blood particles are also highly antimicrobial. And they do and will have applications in the clinic. There are similar materials out there in the clinic using hemoglobin to treat infections topically, certainly in the UK and Europe. If anyone needs to know a little bit more about our microbial particles, antimicrobial particles, the technologies that we hold, then please feel free to contact me, either by email, by telephone, or contact us through our website. The website is shown there at the bottom of the slide. Thank you very much for listening.
I hope you were able to learn more about Veritech's peptide-based antimicrobial microparticles technology from Don's presentation earlier. So once again, if you have any questions for our guest speakers, please feel free to enter them into the Q&A panel, including for Don. We will try to answer as many of them as we can later on during the panel discussion. So for our final guest presentation today, I would like to invite Dr. Lau Kwek Chun, Senior Lecturer at Ngee Ann Polytechnic's School of Life Sciences and Chemical Technology, to share with us his insights to understanding efficacy testing of antimicrobials. Kwek Chun, please.
Thank you, Yaling. So let me just share my slides. So first of all, I want to thank IPI colleagues for inviting me for this talk. I'm Kwek Chun from Ngee Ann Poly. So my talk for today is on efficacy testing of antimicrobials. As we heard from the previous speaker from Kai Yang and also Don, there is a need for antimicrobial discovery because there is since COVID, and also because there's a frequent use of antimicrobials in our environment, and also because there is an increase in antimicrobial resistance. For example, in the hospitals, we see that there is an increased resistance to quaternary ammonium compound. This is often used in the disinfectant and also before surgery and all that. So there are bacteria that has already become resistant to QACs. So to mitigate this problem, new methods have been developed to combat antimicrobial resistance. So these include the physical and chemical methods. And over here, I just want to highlight a few points about the differences between natural and synthetic antimicrobials. As Kai Yang and Don has pointed out just now, the natural antimicrobials can come from plants, animal sources, bacteria or mushrooms, but there is always a concern in some people's mind about the lower yield, right? So if you don't have enough yield, then there's no point in getting this natural source. On the other hand, there are chemically synthesized compound that may be using the chemical formula from this naturally occurring antimicrobial. We can produce them in large scale, so that's chemically synthesized. There are also chemically synthesized compound that goes through modification. So you may have a very good antimicrobial compound, but because the potency may be low or because the yield is low, we can not only chemically synthesize, but we can also chemically modify them to make them more stable, right? Just now, the three speakers has also mentioned about food additives. So natural antimicrobial, if you add them to food, is definitely safer. There are also reports about synthetic or chemically synthesized antimicrobial that has been added as food additive that may be potentially carcinogenic. So using natural is always safer. This is because if you look at it, our liver, our kidneys are the ones that process all the compounds that cannot be degraded, cannot be broken down by our digestive system. So if you take in a lot of these additives that cannot be broken down, then the liver and the kidneys will be the one that has to do extra work. So there's always this concern about toxicity. So synthetic ones, usually they may be, because the way we synthesize them, so it may be harder to break down. So natural ones are easier to break down. So in this case, if a lot of these are released into the environment, either because people consume them or we feed to agriculture or to animals or to fishes and all that, that can lead to resistance in the environment. So there are many different types of antimicrobials that can be found in our environment. I just want to point out to everybody, I'm sure most of us know it already, that there are viruses in our environment, there are bacteria, there are yeast, and there are molds. In our humid environment in Singapore, there are a lot of molds, not just on the food and everyday surfaces, in buildings as well. So those who are working in the marine industry, in aquaculture, they will have to deal with algae bloom eutrophication, right? So in this part of the world, we are not so commonly faced with challenges of parasites and worms, but in different parts of the world, even in animals, you may often find parasites and worms as well. So knowing what microorganism that we are dealing with is important, because then you can select for the appropriate testing methods. So there has been increase in interest in surface cleaning as well as disinfection due to COVID-19. And in Singapore, NEA is the governing body that come out with advisories for testing. So this slide, I just want to point out to everyone that this SARS-CoV-2 over here, otherwise known as COVID-19, belongs to this group of virus called coronavirus. So this group of virus is quite diverse. They include viruses that can infect different types of animals as well as humans. In fact, it's not the first time that we are encountering coronavirus. So in 2003, we have already come face to face with SARS coronavirus, right? So that was in 2003. So in 2019, we had SARS-CoV-2. So in 2020, in less than 20 years, we encountered this pandemic in Singapore. So this SARS-CoV-2 can be tested by different labs, but some other labs prefer to use a safer version. So in our lab, we chose the mouse coronavirus, right? So this is one that is also quite commonly used because it poses less danger for human. In other labs, like Don has mentioned just now, that they use the 229E, and this is also a milder form of the coronavirus. Some of our collaborators are using the porcine version. So all these are viable or safer alternative that the labs can use. Just now, Kayang mentioned about one of the standard tests, EN14476. So ENV also has come out with guidelines that for surface coating, the antimicrobial surface coating, the standard that we are using should be 21702. This is a 2019 method. So basically, this method is for surface coating. So if you have a surface coating like this, we would put in the 0.2 mils of virus, and this is 10 to the power 7 virus per mil. So after that, we cover this with clear film and incubate for 24 hours for 25 degrees Celsius. Then after that, we perform plug assays to determine how much of the virus can be killed. The alternative method that some other labs use would be TCID 50 to measure how many virus particles are left. So in our lab, besides testing the physical methods, we also test the chemical methods. So the physical methods that we have tested include ionizers, UVC, as well as the chemical methods, which is the disinfectant coating, as well as some disinfectant. So we have worked with virus, the MHV, and also the yeast and bacteria. So for bacteria, we use the culture method. So we have gone around different public places in Singapore, as well as the school compound, to look for bacteria. And we have done a lot of culture, and we have also examined the antimicrobial coating to look at how effective they are over a period of time. So the bacteria that we brought back to culture include Staphylococcus aureus, E. coli, and once we have Staphylococcus aureus, we will also further test to see if some of them are methicillin resistant. We have also looked at the total bacteria, the aerobic bacteria, as well as the coliform bacteria. As I mentioned just now, we have also looked at yeast, and the yeast strain that we are looking at is Saccharomyces cerevisiae. So the virus that we are testing in the lab is the MHV. Right? So here I just want to share some of our findings that we have done on high-touch surfaces in public areas, as well as our school compound. Our experience is that non-porous surface, this includes plastic, glass, and stainless steel. When you apply the antimicrobial coating, they are more effective compared to porous surface, which can include wood and fabric. The reason is that for non-porous surface, the antimicrobial coating stays on the surface of the material for longer period of time. And some of this antimicrobial coating, you can imagine them as nano needles sticking out into the air, right? So once the bacteria or virus lands on these nano needles, they get poked or punctured by these nano needles. On the other hand, if you have a porous material, the coating may just seep through and then they get covered by other things. So the effectiveness will be lost in this way. The other finding that we have on the antimicrobial coating is that the effectiveness drops over time. This is not surprising because some of these places that we studied, they are frequently touched by people. And usually when people touch on surfaces, we leave behind our sweat and also our grease and you know, some people may leave behind their food, etc. So this will cover the antimicrobial coating, and over time this coating may also be worn out due to wear and tear, also because people may rub them off and all that. So we also find that when we brought back some of this bacteria for culture, that the vegetative form of the bacteria, that's the bacteria that occurs singly or unicellularly, they are more susceptible to killing than those that form biofilm. Next, I just want to touch on UVC. So we have a project with SingHealth Polyclinic that we are deploying a robot with them to use UVC to clean So our experience with UVC is that it is effective in killing the bacteria as well as viruses. We also tested yeast. In some other literature, they also reported that UVC can kill moles as well. But there are some concerns about UVC in the sense that they can affect human eyes because they are short wavelength. So they can also cause mutation. So when we deploy UVC, we can only do it when there is no one around, right? So in the evening or at night. The other shortcoming of UVC is that because it can only work in using the when there is no blockage. So if you have something that blocks the path of UV, it doesn't work so well. And also, we also tested on different types of surfaces. We found that when the surface is wet, then the UV somehow will be blocked by moisture and all that. So it has to be dry surface and it has to be unblocked. At the same time, we also found that the efficacy of UV drops when the distance increases. So typically, we would say that within 0.5 to 1 meter is optimum. If you stretch beyond 1.5, 2 meters, then the efficacy drops. So over here, I just want to share with you using UVC. When we use UVC on MHV virus for 5 minutes at 0.5 meters, just 5 minutes, we could achieve up to 4 log reduction of the killing, right? So this is equivalent to 99.99% killing of the virus. Similar results can also be seen for bacteria. So after just 5 minutes of UVC, we could see that 99.99% of the bacteria is being killed. We also went around different places to test this ATP test. So basically, this method uses ATP. So the idea behind is that cells, whether bacteria cells, yeast cells, or even human cells, because of the metabolic activity, these cells produce ATP. So high ATP reading means that you have got a lot of cells present. So this is very rapid, and in less than 5 minutes, just swap the surface, and then we just process it and put into the ATP reader. We can get the results in 5 minutes. So it depends on the industry that we are in. So most of the time, say if you are in the food industry and you know that your surface is very clean, right? Or in the hospital environment where you know that the surface is very clean, like let's say for surgery, etc. So if you have some contaminants due to bacteria or yeast and all that, you can easily monitor. So it's more for a test that for surfaces that we are familiar with. But the thing to take note is that this method does not work with viruses. And there are some other factors that can affect the results. This includes the sensitivity of the instrument. And also if there are other chemicals present, this will also affect the results. This I took from a paper. So over here, we see that there are 4 different brands of the ATP reader, and they all have a certain limit of detection. So it depends on which machine you choose. Some machines give a lower limit of detection, but minimally you need to have at least 200 bacteria before the machine is able to give you a reading. Okay, so if you have a surface that is very clean and you have got, say, if you only have 50 bacteria, then you may not register any reading on your ATP reader. Okay, And there is also a higher limit of detection as well. But this most of the time is not of a concern, because sometimes we just want to know if there is or there isn't any bacteria present. Right? This is quite common because it is a chemical base. So when there are chemicals like bleach, for example, right? So I think in this study, they also tested different chemical disinfectants. So this can either enhance or it can quench the reading. So in the most severe case, the machine registered 0 when the actual value is 978 relative light units. Okay, so that's why I mentioned just now that we need to know the surface that we are testing very well. So if you are testing the same surface every day, when there are bacteria growing, we know. But if you are testing new surfaces every time, then some of these factors will come into play, and then they may affect your readings. So to end off my talk, just some take-home message. We need to know what type of microorganisms that we are testing, whether they are bacteria, viruses, yeast, or molds. And also the important thing is, how would the antimicrobial be applied in everyday setting? Are you going to use it in food? Are you going to use it in surface coating? Are you going to use it for personal use, for cosmetics, right? So then the testing method that we will be using will be different. The last point that I want to share is this thing called the Accredited Labs. So if you go for the accredited lab to do the testing for you, it will be more expensive, but the advantage is that when you send your results overseas, they will be accepted internationally. So for non-accredited labs, they would be cheaper, right? So most of the labs that you find in the IHLs, they are non-accredited. So the commercial labs are usually accredited. Some of the accreditation includes the ISO 17025. I myself work with SAC Singlass. That's also part of Enterprise Singapore. So I go around to the national labs to do audit as well. So accredited labs are usually more well-equipped, and they can deal with international methods, right? But they are more expensive. So it depends on your application. What do you want your results for? So for most of the time for screening, right, you may want to go for the non-accredited labs. But after you've done your screening, you may want to send your product for accredited lab so that you can sell it to overseas market. Okay. I just want to acknowledge my colleagues in Ngee who has been helping me with all this work, and also our collaborators in NUS. Prof. Vincent Chow is my teacher as well as my good friend and now my collaborator. Prof. Pablo is an expert in antibiotic-resistant bacteria. So we also work closely with SingHealth Polyclinic, Prof. Tan and Dr. Yusrina. We also want to acknowledge our funding bodies, MOE, as well as Temasek Foundation for supporting our work. Thank you very much.
Thank you, Craigton, and again also to Kayang for your presentations. So the last segment of today's program is a panel discussion with our guest speakers, moderated by my colleague Paula, Technology Manager from IPI Science and Engineering team. So may I invite Paula and also our guest speakers to turn on the video? Right, yeah, on. So Paula, over to you.
Thank you, Yali. So good afternoon to everyone in the audience. Thank you once again to Kayang and Craigton for your presentations on the plant-based antimicrobial derived from food waste and on the intricacies in efficacy testing of antimicrobials through the various projects you've worked on. So As Yaling has mentioned earlier on, it's a pity we're unable to have Don join us live today for this panel discussion. But let's proceed with Kayang and Kwek Chun, who will be sharing with us more insights and knowledge on antimicrobials. So to kickstart this session, right, I think the first question, let's direct this to Kayang. Can you comment on the durability of Ultra Bactec? For instance, like how long the antimicrobial function can last, since this was really not covered explicitly during your presentation. Then there is also a question from our audience on whether you have tested Ultra Bactec as an antifungal agent and whether it's an inhibitor or bacterial eliminator.
All right, thank you for the question. So maybe let me answer it one by one. So for the first question on the durability and stability of the Ultra Bactec, currently we are doing a lot of tests and in terms of accelerated shelf life as well as the stability test when being applied. So in the instance where we apply our surface disinfectant to a certain type of surface, so we just leave it there for different time intervals from 30 days, 60 days, and even 90 days. And then we add on bacteria to it to see how does the surface react to that bacteria. So what we have shown is that our Ultra Bactec is able to be stable on any surfaces for as long as 90 days. Currently, our time point is until 90 days. And even after 90 days, when we add the bacteria to that surface, which is sprayed with Ultra Bactec, it is still able to kill the bacteria that we have inoculated 90 days later. So the stability is definitely there, right? With regards to the second questions on the fungal, yes, we do have, we have shown that it is efficacious against fungal, just that it is a bit different because as what I have shared, the antimicrobial functionality or the mechanism basically works by targeting the cell membrane. That's one of the main mechanisms. And the membrane of fungi are basically more complex, and hence, when you are trying to target things like fungal and yeast, you basically needed a higher concentration of certain compounds to elicit a more pronounced effect, right? Yeah. So the third question on whether is it an inhibition or is it an elimination, right? I can confidently say that the Ultra Bactec works by an elimination method, where when we test the minimum bactericidal concentration, we really plate all the suspension onto a certain agar plate. So we do not observe any growth of the bacteria. So it is definitely not inhibitory, but elimination.
Okay, thank you. So our next question is actually for Kwek Chun. So you know that antiviral tests can actually be very costly, right? So how do you think companies can mitigate this during their development process? And also, to take in one of the questions from our audience, right? You know, how can we make consumers aware, you know, of these antimicrobial claims, right, without creating a wrong perception? You know, all the productory claims, you know, on, like, say for instance, the disinfectant sprays that we can get from our supermarkets, or even the hand sanitizers, things like that.
Thank you. Thanks for the question. So with regards to your question on antiviral tests, yes, the test is expensive. Reason behind is that it's not as straightforward as just applying your antimicrobials onto virus, because virus itself don't grow and it's different from bacteria or yeast cells. So viruses are basically intracellular parasites. So in order to see whether your virus is there, we need to have a suitable host cell. So this involves a lot of screening beforehand. So knowing your virus is important. So, and also we need to test to see which method is the best to achieve the results. So if let's say I want to have 99.99% killing that I want to test, I need to have a high enough virus load to begin with. So if I start with say 10,000 virus, at the end, if it's 99.99% killing, I may end up with just a few hundred virus. So that's, or even one virus left, right? So that's a 99.94 log reduction. So in order to achieve this, you know, that high percentage testing, I need to grow virus first. So that involves a lot of effort on the labs and all that. That's why it's expensive. So to answer your question, how to reduce the cost,
Yeah.
I would say maybe start with the non-accredited labs first, as I mentioned just now, or you can do some collaboration. Say if you have got a good project or good product, you may want to do collaboration. So in Ngee we are open to collaboration. It doesn't mean that everything you have to pay us to do the work, because we also want to help industry and all that. So we are open to collaboration as well. So your then after you have selected your product that you are comfortable with this product or and all that, then we can go for the accredited lab so that your results can be immediately recognized overseas. Say if you want to sell your product overseas, and you can apply for the license to sell overseas. Yeah, that's the first step. So with regards to your question on awareness of antimicrobials, I understand that there are a lot of claims and there's a lot of false claims as well. So as I mentioned, you need to know, if possible, read the fine prints as well. Sometimes we see a lot of claims online, even, not just in the supermarket, that this solution can be antivirus, can be anti-everything, but we know that is not possible. So I would look very carefully as well what bacteria or virus they test. So recently I came across one product that online, and they say can kill coronavirus, can kill SARS-CoV-2. But when I read the reference, they are just using bacteriophages to do the test. So it's quite different. Bacteriophages are basically virus that infects, viruses. So they are nowhere close to human viruses. So I think because of the awareness of virus, a lot of people are also interested to know more about viruses and all that. So again, in Ngee we do conduct short courses on viruses and bacteria. We even conduct short courses for secondary school students as well on microbiology to get everybody to be more aware of the microorganisms in our everyday life. So we are all doing different part to increase the awareness. I think IPI is also doing a good job in promoting this by having us to give the talk.
Thank you. I think the importance of product validation, right, when it comes to antimicrobial hormones, is definitely important for companies to consider.
Yeah.
So moving on, I want to pose this following question, right, to Kayang on antimicrobial peptides actually. So, in Don's presentation, he covered on, antimicrobial peptides briefly, right? And since you already have some experience working with such compounds, we know that these are, charged molecules more commonly used in clinical applications, such as, like skin and wound healing therapeutics and as food preservative, right? So, they have a lot of structural and functional diversity. So what other non-health and non-food applications, say for instance, can they actually be used like in coatings or for like master batch processing on porous surfaces such as textiles? So just some of your thoughts about this.
Sure. Thank you, Paula. Wow, this brings me back to my old days when I'm doing my PhD times, when I work with antimicrobial peptides. Antimicrobial peptides are interesting because inherently they are found in all mammals. It's just that when we do a lot more modification, we make sure that these peptides are short, easy to produce, and cost efficient. So they are very effective, yes, but there are limitations to use of antimicrobial peptides because, again, they are protein made out of amino acid. They are short protein, so they are very susceptible to certain treatment conditions like thermal treatment, pressurized treatment. So to say that, right, as long as your process doesn't have such kind of a harsh processing condition, antimicrobial peptides are generally quite good, right? I have seen antimicrobial peptides being used out of biomedical sector. It can be used in cosmetics. I think a lot of cosmetics have a certain extent of antimicrobial peptides. We have also tried during my PhD days. We have also tried to put antimicrobial peptides into certain type of coatings, polymer coatings, where they have a certain release over a certain period of time. So it is possible, and the use of antimicrobial peptides is definitely that there are a lot of potential for its use.
Okay, thank you. So I think let's move on to the next question, right? So perhaps to both of our panelists present here, right? So given that, this webinar's focus is really on organic antimicrobials that are naturally occurring, so what are your thoughts on such technologies, right, against synthetic compounds like chlorine-based agents, metal particles, quaternary ammonium compounds, and, nanoparticles like TiO2? How can such natural antimicrobials compete against these synthetic versions of it, right, that are commonly used in the market? Perhaps let's start off with Kaya.
Yeah, to me, I think
Sure.
like our company's philosophy, right, is nature always gives us the answer, right? Which is true, right? When we talk about things like antibiotics, the first antibiotics, penicillin, we discovered it from penicillium, that fungi, right? And then we evolved and use science to develop more and more antibiotics. So I do think that the potential of natural antimicrobial is definitely huge. It is just whether do we search hard enough, and that is where our company set out to really source and find really de novo natural biomolecules that is present in nature to use to see whether can they kind of like replace or complement the current synthetic chemicals.
Okay, so Quek-Chun, perhaps you'd like to chime in next on this?
Sure. Thanks. I agree with Kayang that he's putting his effort into developing organic antimicrobials. I salute him. So it's a tall order, but I think he succeeded. So taking the example that Kayang gave on penicillin. So penicillin was originally from fungus, right? So during those days, the yield was super low. So they couldn't extract enough from the fungus. So that's from natural. So in fact, when Alexander Fleming got the Nobel Prize, he shared it with two chemists. So these were his collaborators who helped him to extract them using chemical method. And later on, penicillin was in production. So it was chemically synthesized, right? So chemical method can help, but again, the future modifications, either you add the side chain to your penicillin and all that made it more stable and make it more potent. These also make the penicillin more resistant to being broken down naturally. So as a result, due to the frequent use nowadays, penicillin, even the derivative of penicillin, they become a lot of drug-resistant bacteria are not scared of penicillin anymore. So I think there's a challenge there, and also the pharmaceutical companies, they are not producing new antibiotics because every time you come out with new ones, there will be frequent use, and then the cycle begins again, the resistance starts. So I think natural antimicrobial has an advantage because they are easy to break down. So unlikely that they will cause antibiotic resistance or antimicrobial resistance. And also sustainability-wise, it's more sustainable, right? So you don't have to use a very costly method to produce. And also they are more environmentally friendly.
Yeah. Okay, thank you for your thoughts about that.
Yeah.
Right, so moving on to the next question, right? So actually through our engagement here over at IPI, one recurring concern that companies have when incorporating such antimicrobial agents into their products are actually regulatory considerations to undertake. So we know, for instance, that over in the US, right, there is EPA that regulates this. In Europe, we have the ECHA. So over here in Singapore, there are currently no firm regulations for non-human use applications, right? Except for the guidelines and recommendations that Quek-Chun has actually, showed from the NA website. So I guess to both panelists again, so what are your thoughts of, this type of regulatory aspects with respect to the antimicrobial products found here in Singapore? And could you perhaps also share your experience when working with your partners or companies regarding, all the regulatory considerations? Maybe Quek-Chun first?
Sure. Thanks for the question. As far as I know, there is no regulation on use of antimicrobial on animals. So potentially there is a market there. So in fact, a few years ago, we worked with a company that wanted to sell the his line of antimicrobial to pet owners and all that. So I think there's a market. So the only thing is that how do you sell it to them? You know, if you don't have that scientific backing. So I think in that sense, we provide the scientific data to back up his claim, and we also help him to test what kind of bacteria the antimicrobial can be effective against, especially for animals. They may have different types of bacterial infection, etc. I think it all boils down to whether you know the market, right? So you can come up with a very good product, but in the end, nobody buys or nobody believes, then it's also useless. So that's why I say I salute Kayang, because he's doing a good job, not only doing the science part, he also needs to do the marketing.
Yeah. So perhaps Kayang, you would like to chime in here?
Sure. So basically, I would think that, like what you say, right, in US and Europe, there are regulatory bodies regulating this, but in Singapore, currently we do not have this. So of course, I believe that the government agencies are looking at this closely, especially with more and more frequent use of antimicrobials. So as a second line of defense, right, normally what we have to do as corporates, as businesses, is really to do our due diligence, to go through all the rigorous testing, and as such, right, so all the testing reports and all the testing reports that they pass and they meet certain criteria, it becomes very important, right? And that's why we are putting in a lot of effort and investment to do this, to make sure that we are really selling something that is efficacious and do not harm the general population as well.
Okay, thank you. So I believe that's all the time that we have for today. So thank you once again to Kayang and Quek Chun for actively participating in this session. And now back to you, Yale.
Thank you.
Thank you, Paula. And now we have come to the end of this webinar. So once again, a big thank you to our speakers and also to all our attendees for taking the time to join us today. I know we have overrun a little bit, but I'm glad that you stayed on with us. So we hope that you have really benefited from the various presentations and also the interesting panel discussion.